Assalamu alaikum students. Today we will study about the cell and cell division. But before watching this video, please subscribe the channel.
Overview of meiosis. Parent cell with diploid number of chromosomes divides into 4 daughter cells with haploid number of chromosomes. So that's why meiosis is also called reduction division, as it reduces the number of chromosomes to half the number. Why? One cell divides into 4 daughter cells because, for example, in this case, the number of chromosomes in the parent cell is 4. So after the DNA replication, the number of chromosomes, they get 8, and these 8 chromosomes will be divided into 4 cells equally. 2 chromosomes would be going to each cell. Meiosis takes place in 2 stages, meiosis 1 and meiosis 2, that we will discuss right now. This is the start of meiosis 1 with its prophase 1. Early prophase, we see that the DNA has been replicated. Centriole divides into 2. The 2 centrioles move towards the opposite sides, and the spindle fibers, they are being made by the centrioles. In the late prophase, we see that nucleolus disappears and the cell membrane also starts disintegration, and the spindle has been made fully. The homologous chromosomes, which are the chromosomes which are similar to each other. In the homologous pair of chromosome, one member of the pair comes from one parent and another member of the pair comes from another parent. The homologous chromosomes, they start pairing up along their length, and this pairing up of homologous chromosomes is called synapses. The homologous chromosomes, chromatids, overlap each other, and this X shape is formed, that is called chiasma. When these chromatids of homologous chromosomes overlap, this process is called crossing over, and during crossing over, the chromosomes chromatids exchange their parts. For example, you can see the red chromatid has a little part of blue attached to it, and the blue chromatid has the little part of red attached with it. This exchange of parts helps in the recombination of alleles or genes. Genes means the segments or parts of chromosomes. These segments or parts of the chromosomes, they are responsible for different characters of the organisms. So in the prophase, late prophase, crossing over takes place by pairing up of homologous chromosomes, and then next phase of meiosis 1 starts, that is called metaphase 1. In the metaphase 1, the homologous chromosomes, they pair up on the equator of the spindle, that is the middle part of the spindle. The homologous chromosome pair up, and then comes next phase, which is called the anaphase. So the homologous chromosomes, they start moving from the equator or from the center to the poles of the spindle. So as we discussed, that one homologous member of the pair will move toward one pole, and the another homologous pair member of the pair will move towards other pole. So when they have gone to the poles, the anaphase finishes, and then comes another phase of meiosis 1 that is called the telophase 1. So in the telophase 1, the nuclear membrane reforms and cytokinesis 1 takes place by the cleavage of the cytoplasm, and it will result in the two cells with two chromosomes, but the two chromosomes are having the two chromatids. This is the start of meiosis 2, and here we see the prophase 2 in the two cells made by meiosis 1. We see in the two cells the cell membrane being disintegrated. The centriole divides into two, the two centrioles moving towards opposite poles and the spindle fibers being made. The ester has been formed around each centriole in both of these cells. And after that comes the next phase of meiosis 2, that is metaphase 2. Here in the metaphase 2, in both of these cells, the chromosomes arrange themselves in the center or on the equator of the spindle. We see, and these chromosomes, they are now arranging themselves in a single file. After that comes the next phase, which is called the anaphase 2. Anaphase 2 starts with the division of centromere in the center of the chromosome. The centromere divides and the chromatids of the chromosomes, they get separated, and the spindle fibers contract and they move the chromatids towards the opposite poles. This chromatid will move towards this pole and this chromatid will move towards this pole of the spindle. In both of these cells, this anaphase 2 takes place by movement of the chromatids towards opposite poles after the division of the centromere in the center of the chromosome. After anaphase 2 starts another phase, that is telophase 2. Telophase 2 means the nuclear membrane around each chromosomes, around each set of chromosomes, it is reformed, and it is followed by the cytokinesis 2, that is the division of cytoplasm in both of these cells. So after cytokinesis 2 in both of these cells, the two cells made by this and two cells made from here. So in this way we will have 4 haploid cells, and these 4 cells are with nucleolus and the nuclear membrane and with half the number of chromosomes, because these are the gametes. The importance or significance of mitosis is important for growth and development. Growth means increase in the number of cells, and development means when the cells differentiate into different tissues or organs. Mitosis is also important for repair of worn out tissues. For example, the cut skin will heal up, or the fractured bone cells are going to regrow. Mitosis is also important for asexual reproduction. For example, some plants like potato, onion or garlic, they reproduce asexually. But if error in mitosis occurs, the cells will keep on dividing, and this will result in the lump of cells or the guilty that will appear in the form of disease called cancer. So importance of Another kind of cell division, meiosis. It is important for sexual reproduction, and it is also important for restoring the normal number of chromosomes in the body cells. But if errors occur in meiosis, obviously this will result in the abnormal number of chromosomes in the body cells, and it will appear in the form of hereditary disease. For example, Down syndrome is a hereditary disease, and the individual with Down syndrome, they have 47 number of chromosomes instead 46 number of chromosomes, which is the normal number. Why this 47 number of chromosomes in the individual with Down syndrome? Because the error occurs in the female gamete, and the 21st pair of chromosomes, the whole pair goes in the egg cell of the female, and when this egg cell with 24 number of chromosomes instead of 23, it combines with the normal male gamete with 23 chromosomes. So 24 plus 23, this will result in the 47 chromosomes in the individual with Down syndrome, and these individuals are with the disease, and they are not behaving like normal people.